Welcome to an introduction to clinical research. My name is Paul de Koning. During this presentation, I will cover the history of clinical research, how regulations on clinical research came about, what are the causes of the low and declining probability of success in pharmaceutical R&D, what we can do about this, how inroad of biomarkers has helped somewhat in reversing this low probability of success, and how OEDES may help you. The history of clinical research goes back to 562 before Christ, where King Nebuchadnezzar, who ruled Babylon, allowed soldiers who wanted to try a vegetable diet to do that for 2 weeks. The common diet at the time was meat and wine, and one of the officers had suggested that following a vegetable diet would result in more healthy soldiers. After the 2 weeks, the soldiers that had indeed followed the vegetable diet looked more healthy, and King Nebuchadnezzar allowed them to continue the diet. In 1747, there's a record of the Scottish surgeon James Lind, who was a doctor on a UK army vessel, who did a trial to try and reduce the effects of scurvy. He took 12 sick people with scurvy and divided them in 6 groups, and by pairs they would receive either cider, seawater, vinegar, vitriolic elixir, a purgative mixture, or 2 oranges and a lemon, and only those that received 2 oranges and a lemon they recovered from scurvy. The first more controlled clinical trial was conducted in 1943 by the Medical Research Council in the UK. It was the first double-blind study of a penicillin-type product that was administered as a solution every 4 hours in the nose of those with a common cold. They did that for 2 days, and then at the end of the 2 days it was found that those that had received the penicillin-type solution did somewhat worse than those on placebo. In 1947, again, the Medical Research Council of the UK conducted the first randomized double-blind study of streptomycin tuberculosis. This was probably the first example of a real well-controlled study, although no informed consent was used before the subjects entered the trial. The regulations on clinical research actually came about each time and were updated continuously after issues occurred. For instance, in 1851 in the UK, the Arsenic Act came into effect following the poisoning of people by arsenic acid, not even accidentally, but deliberately as a way to murder people. In 1938, the FDA actually stipulated the Food and Cosmetic Act following the poisoning of children and the death of children after they had received a sulfanilamide preparation where diethylene glycol was used to dissolve the product. In 1962, some of you may recall the Softenon affair, where children were born with almost no limbs, and that resulted in legislation by the FDA where next to looking at safety, it was now also necessary to look at efficacy. For a long time, there was a sort of a standstill in the regulations until 2006, when I recall vividly, I woke up to the news that in the UK, in a hospital, healthy volunteers that had participated in a trial of a CD28 agonist antibody all ended up in hospital after they developed a sort of cytokine storm that led to organ failure. The upshot of that particular trial was that from then on in first-in-man trials there would be sentinel dosing, so there would be 2 volunteers only dosed, 1 on active, 1 on placebo, and one would wait and see how those two would do before the rest of the group would be dosed. This new legislation on first-in-human dosing was even further updated after in 2016 in Rennes in France. One volunteer died after, in a trial, a multiple dose trial of a FAAH inhibitor. Actually, this volunteer was taken to hospital on the evening on day 5 with the symptoms of a stroke. And despite that serious adverse event, all the volunteers were dosed the next day, so on day 6, and all of these volunteers again were taken to hospital with symptoms of a stroke, and some of them died. With regard to ethical issues, the start of the definition of ethical principles was in 1947 as a result of the Nuremberg process, where the atrocities of the Nazi regime and the doctors who actually did what they called research during that time were trialed. This eventually led to the Declaration of Helsinki in 1946 that set out the ethical principles for medical research involving human subjects. Despite all of these sort of rules, principles that doctors need to abide by, things continue to happen, like in 1997 when there was a trial of AZT in pregnant women in South Africa, where they tried if a lower dose than the well-established effective dose of retrovirus would also be protective in reducing the transmission of HIV to the unborn child. And these women received either a low dose or placebo. The upshot of that trial was that it was universally agreed that every subject in a trial from then on should have access to the gold standard at least. The EMA, the European Medicines Agency, for instance, stipulates now that all trials that are conducted outside of the EU should guarantee that the participants receive at least the same standard of care as one would receive in the European Union. Quality issues are actually a major issue and very often actually the reason for non-approval of drugs. A famous example, unfortunately famous, is that in 2012 there was a multi-state meningitis outbreak due to a mold contaminating a compounded sterile preparation that was used for multiple sclerosis. And in green there on the map of the United States you see where the victims of that preparation lived. All of the regulations around the conduct of clinical research are summarized by ICH in the E6 guideline on good clinical practice, and actually you should look at revision number 2. So it's a set of guidances and principles to achieve greater harmonization worldwide to ensure that safe, effective and high quality medicines are developed and registered in the most resource efficient manner. So we've seen since the end of the 1990s that the probability of success in the pharmaceutical R&D has gone down, and actually dramatically. And there are a number of reasons why that is. The first one is the level of target validation. Until that time, until the 1990s, pharmaceutical industries would work on the approximately 300 well-established targets. These were targets that were well described, and each of them, for instance, had at minimum 300 publications. So it was about the function of a particular receptor or enzyme or protein. Nowadays, Much less is known on the very many recently identified, often genetic targets. There might be a few or even no publications. Disease models, it should be recognized that the typical mandatory animal toxicology studies are no more than 70% predictive of human toxicology. So if you look at the mandatory 2, 3 or 6 month results of toxicology studies, they do not predict more than 70% of what might happen as safety issues in humans. With regard to the so-called animal disease models, only about 1/3 translate into human efficacy, and that holds also for the traditional oncology models. Preclinical research, which often, of course, is the basis for starting clinical research, was, as was found out in the early 2000s, not always as rigorous as one would have hoped. It was found out that the results of about 50% of preclinical research studies in top journals cannot be repeated. These preclinical animal studies are not conducted with the same quality and rigor as human studies. For instance, they aren't repeated, often not double-blind, placebo-controlled. The clinical research, there's often a reliance on dose-limiting toxicity, for instance, in oncology trials, of the 3+3 design as a means to establish the optimal dosing for phase 2 and 3. The result is that dose is often not optimal. Also, there's a failure to establish target exposure, human pharmacology as a means to confirm proof of concept. And also by pharmaceutical companies, there's too much hope that, well, we've seen it works in this tumor, so it will probably work in that tumor as well. Let's do a phase 2-3 trial rather than start with a phase 1-2A trial. So what can we do about this low probability of success? We can try and improve the target validation, try to humanize preclinical research as much as possible, use patient-derived cells, organoids, microphysiology systems, network medicine, so combining systems biology, systems pharmacology, and systems toxicology to understand more about the actions of a particular target, where it sits in the cascade that ultimately leads to disease. With regard to preclinical research, we can try and enhance reproducibility of preclinical research by repeating experiments and do them in a double-blind fashion, and implement quality guidelines on aspects of material, study design, data analysis, reporting, and lab protocols, just as the rules around clinical research. There's a special role for exploratory clinical research, and there's a special training on that. It's something that came about in the early 2000s as a way to try and take earlier decisions using a combination of biomarkers and routine clinical endpoint in order to see whether the invention that preclinical showed so much promise actually has likely added clinical benefit. Other things that we can do is to try and optimize the choice of the efficacious dose using all data from an early oncology trial rather than only the dose-limiting toxicity in 3 patients, and to also better use biomarkers. And again, there's a special training on biomarkers. Oncode actually is a very good example of collaboration between academia and the government, but also there can be even wider collaborations between academia, pharmaceutical industry, the regulators, the payers, the prescribers, and the patients in order to all agree on what is really needed, what is the definition of unmet medical need, and when will something be called a real benefit. So biomarkers, it took until 2001 until one came up with a definition of biomarkers, which reads, it's a characteristics that is objectively measured and evaluated as an indicator of normal biological processes, pathogenic processes, or pharmacological responses to a therapeutic intervention. Biomarkers have long been used as a sort of surrogate marker, for instance, in clinical decision making, like blood pressure was used as a surrogate marker for ultimate reduced cardiovascular events, just like cholesterol lowering, or the reduction in HbA1c in the management of diabetes was used as a clinical endpoint for registration and not the ultimate deleterious effects of diabetes and how a new drug could reduce those. Like progression-free survival is currently used as a predictor of overall survival in oncology. Since the early 2000s, drugs are sometimes developed together with a biomarker that predicts actually whether that new drug will work in a particular patient. It's part of the precision medicine initiative. And when a biomarker is developed as an integral part of the development of a new drug and registered together with that new drug, it's called a companion diagnostic. Besides that one-on-one biomarker drug tandem, there are also many efforts to predict prognosis or response using, for instance, gene signatures. And although many of these have been developed, very few of these latter platforms have actually received regulatory approval and are reimbursed by insurers. For all biomarkers, there needs to be a level of validation. Scientific validity, does the science support the association and the use? Analytical validation, for instance, reproducibility, sensitivity. Clinical use validation, for instance, positive and negative predictive value. Do clinicians trust the test to inform clinical decision making? Validation is an iterative process with more stringent norms and requirements in later stages, as the impact of false predictions is more serious and as it impacts larger populations. So you get a sort of fit-for-purpose validation. The more consequence the use of the biomarker potentially has, the better the validation should be. So what is OEDES? OEDES is the Oncode Exploratory Development Expert Support Group. They have been established by the OEDES management team as a way to help Oncode investigators make the transition from the research phase to the clinical development phase, to help define clear project goals, to help optimize the development path, to support the definition of early decision criteria, and to form an expert panel that can be consulted. So on the right-hand side of this slide, you see the core team members of the OEDES group. Sophie Gerritsen, who is a clinical scientist, Emil Pot, who works in business development, Ellen Hulscott, who has a long experience in clinical trial design, Danny Berg, who is a toxicologist, and myself. So how might we help you? We can, in the very early stages of research, support you by having a quick discussion on what it might take for your new invention to go to the clinic. Or we might help you in submitting a research plan to a pharma company seeking a collaboration, for instance, requesting material. In the CPOC phase, we can support you in preparing and reviewing the expression of interest. We'll usually hold a on-site or currently digital workshop to help define the fundamentals of exploratory clinical research as a basis for successful full proposals. So how do you define that early clinical development phase in a way that leads to early decision making? And we can review your full proposal and assist in answering to required amendments. In the phase of clinical research, we can provide assistance in preparing for a proof of concept decision, and we can provide a sounding board and offer possible solutions when unforeseen issues surface. On this slide, you'll see actually a linked library to relevant publications and guidelines in the area of clinical research. I hope you find this presentation useful.